Hello, good morning, everyone. I'm Keshav, like Tanweer mentioned. So today I'm going to talk about, before I start that, I thank the organizer for calling me, and it's a nice meeting. And I'm happy to present here because we continue this meeting every month in our biological division, where we call it as RNA group meeting, where most of us today we are presenting, or actually we meet almost every month in a once there to talk about our works. So today I'm going to talk about a protein called DDX3X. It's a pretty interesting protein because it's called DDX3X because it's a X-linked gene, and it has a homologue called DDX3Y. Essentially, this protein is expressed in almost every cell you name it, but DDX3Y is stick to only germline cells, especially where Y chromosome is there. So before I go into the talk, and these are the people who actually did the work, and I'm just presenting their work. So Paulomi, Shivani, and Savita are from my lab, and Shivani is the one who was presenting poster here. And we have collaborators, Raj, who's one of the organizers, and her student Swati, who is helping us with polysome profiling. And I have a long-term collaborator from University of Hyderabad, Prasad, who helps us with neurobiology. I think there are many talks for the last 2 weeks which were trying to emphasize more on how RNA binding proteins have a big role in deciding neuronal cell fate as well as neurodegeneration. For example, if you see all these TDP43, E3BP FOS, and including DDX3X, typically many of these proteins are known to condensate into a liquid-like compartments called stress granules. In general, if you look at a cell, and these stress granules are assembled in order to withstand the stress so that they arrest the translation and they freeze the cell in a survival state for some time. But if this persists, and cells anyway will undergo cell death. But essentially, these stress granules are like some kind of a buffering system to hold on to the stress for some time. And then, like many people might have presented already, that they disassemble when the stress goes off, and then the functions will rejuvenate. And we work on this protein called DDX3X, and it's pretty interesting protein because it's conserved from yeast to all the way mammals and highly similar. And they have flanking NN tunnel intrinsically disordered regions, and they have a core helicase domain which has 2 Recke-like domains. And its functions have been very well established, but not at the level of organism because you can't create knockout mice. And even you can't create a mutant mice, it's very difficult to do that. So it's been known to be associated with unwinding complex RNAs during translation initiation, and it has been associated with some innate immune sensing of RNA molecules, also known to be associated with Wnt signaling pathways. And this is the protein, DDX3 protein, which has 2 domains called D1, D2, which is associated with the RNA over here. So I'm not an expert in phase separation, to be frank, but we came into this field for a reason. So when I was working in postdoc, we discovered a unique function to this DDX3X protein in deciding immune cell fate decisions. So one is, like I mentioned, DDX3 is a protein which can go into a stress granules. And it's also part of the important protein to assemble stress granules like G3BP1. Okay? And that too, it happens because of this intrinsically disordered regions in the immune cells. Okay? And not only that, it also participates in one of the cell death pathway called apoptosis, which is an inflammatory cell death pathway. So it's a bit of a different from apoptosis because here everything is opposite to apoptosis. So when this particular pathway is activated, there are proteins called pore-forming proteins. They get activated, go to the membrane and form pores, and then cell will die by lytic pathway. So essentially all intracellular contents come out and then activate inflammation, chain of inflammatory. So we discovered that this protein DDX3X is important for assembling this complex called inflammasome, which is required for activating these pore-forming proteins. These inflammasomes are large complexes. When they assemble in the cell, they vary in the size from 2 to 7 microns in size, and then they eventually activate this lytic form of cell death and inflammation. Okay? It's pretty interesting because it's something people always thought that this protein has a role in translation or RNA unwinding function, but this is something that we come up. And then we try to understand that how does it work between stress granules and inflammasome. So we proposed a mechanism in this paper, I'm showing that that if DDX3 first try to promote activating stress granules or assembling stress granule, its availability is limited for activating inflammasome. So it only retains the cell survival. But if DDX3X activates inflammasome, it particularly activates inflammatory form of cell death pathway, and the cell survival is not retained. Okay? But how this DDX3 makes the decision of which pathway to activate is which we still don't know, and we're trying to put some effort on that. So I just put catch me if you can, because whoever catches DDX3X, and that goes into limelight. Okay. So the two pathways that I was talking about, liquid-like compartments or stress granules or inflammasome, they're very well implicated in many of the neurodegenerative diseases. You just type the name of these things with the neurodegeneration, you'll find many papers on that. And in particular, I would like to indicate inflammasome. Okay, this is the paper from Michael Henniker's group, where they try to see that the inflammasomes, here inflammasomes are labeled with ASC, they act like nucleating factors for cross-staining beta amyloid. It's very well established. Now, even like, for example, if you delete the inflammasome components, the aggregations like tau and even amyloid aggregations have been reduced in this in the mice model. Okay? The reason why I was pointing out this data is that now we found that DDX3X is nucleating factor for this inflammasome complex itself. So that's why we wanted to understand that how this DDX3X is somehow linked to the neuroinflammation and neurodegeneration, because it's kind of a master regulator that can promote two different signaling pathways or large complexes that is directly associated with the neuroinflammation and neurodegeneration. But at homeostatic condition, DDX3X tend to regulate them so that they don't activate abruptly. But what happens in the disease condition is something that was not studied. So there is a disease called DDX3X syndrome. Okay? The patients with these mutations in DDX3X protein tend to show a variety of symptoms, particularly in neurodevelopmental disorders and cognitive functions. It's kind of an interesting thing for us to start with, because we wanted to understand DDX3 role in the neuronal conditions. And typical features of them is having polymicrogyria or microcephaly, where their gyri has been like, their morphology has been stained, and they eventually end up like having autism-like symptoms. So this is where we started. We wanted to understand, and there too many mutations have been reported by now. Earlier it was thought that it's been only seen in females, but there are few mutations which now have been reported in the males as well, kids. Okay. Since a lot of mutations, we wanted to take few mutations, that to missense mutation, because it's difficult to characterize. We had a structure-based approach to finalize few mutations so that we can put bets on them and start understanding how they are affecting. So we particularly looked at the mutations or selected the mutations which either could be at the interface of RNA binding, or they might be in the protein, but they are buried and they can disrupt if they are mutated, something like that. Okay? So these are the over 10 mutations that we have selected. And the mutations that we have selected over here, some of them have been already implicated in associated to the cancer progression too. Say the same position, but the mutations may be differed, but the position appears to be very important, that anything that comes to a change that can bring a pathological thing. Not only that, we used this Google DeepMind thing called alpha missense. It's basically this alpha missense, what does it do is that if you take any protein and give it to that, it tries to predict the pathological consequence of mutating a particular amino acid. For example, you have P40. If P40 is mutated to all the positive amino acid, what could be the detrimental effect of this protein function? And it is combined with protein folding prediction as well as clinical variation score, and it takes a lot of parameters. It's pretty robust method. And you can see that the mutation that we selected, all of them, the green ones, tend to show a high pathogenicity score. And the blue ones are the cancer mutations, even they tend to show a pathogenicity score, which kind of gave a confidence to go ahead with these mutations. So we wanted to start understanding how these mutations can impact the DDX3X function, right? So we started to see that how these mutations would be affecting DDX3X phase separation function or stress granule function. So that's why we scored for stress granules. In this case, we started with HeLa cells. before I go into the data, I wanted to say that we tried to create HeLa cells, which are where we wanted to knock out DDX3X gene. We did create the knockout cells, but those cells don't persist in the culture. They tend to die with time. So we could not have a clean system. So we went with ectopic expression or overexpression system. But you can see that it's a huge data, but focus on here. We can see that when we try to induce stress granule with the sodium arsenate, and some of the mutation tend to form stress granule which are high in number, like they go way high in number than the wild type thing. And this phenomenon we observed in other different cell type also, like neuro 2A cells, which is a typical neuronal cell type from that. And some of these mutations, they tend to show increased stress granule formation in them. And then we asked that why DDX3X mutations are favoring phase separation function of DDX3X. And then also we wanted to see that does these mutations can impact the phase behavior of this DDX3 protein, because it's been known that if you change the liquid property of stress granules or phase separated granules into solid-like, they behave completely different in their function-wise. So we established a simple assay where we take cells, we induce stress granule where DDX3 co-localize and wait for some time, then wash off. Ideally, they tend to dissolve. Like you can see here, these stress granules are induced in where wild-type DDX3X expressing. You wash off, they tend to go off. And then now, in this case, now look at how the mutations are behaving. One of the mutation I represented here, it tend to retain the stress granule formation. That's what we tried to show, that we scored this in two different cell lines, both HeLa and neuronal 2A cells, where we see that some of the mutations, despite we wash off, they tend to retain the granules for some time in the cells. Which is kind of an indication for us that these DDX3X mutations favoring the stress granule, which can persist in the cell for longer than usual. And then we wanted to understand more about them to see that what if there is a change in their liquid-like properties to solid-like. So we took a state-of-the-art method, FRAP, where now here you can see that this FRAP for wild type, once you put the wild type, once you shine the laser on that, it'll go off, then it'll come back because of its liquid-like behavior. But the mutant, if you look at mutant, once you flash laser light on that, then the signal will go off and it will never recover from that. So that kind of indicates the liquid-like or solid-like behaviors, and then you can tend to plot that and see that how does those curves look. So here you can see that the wild-type protein, so once you put the laser on that, it loses its fluorescence and then tend to recover that. But there are majority of the DDX3X mutation, they don't recover fully after FRAP. And this is not only case with HeLa cells, and we even tried in neuro 2A cells. We tried these assays in two different cell lines for a reason, because we wanted to see that it's not a cell type specific phenotype. And we did find that there's one mutation called 480, if you can see. In HeLa cells, it tends to show that it doesn't recover. But in the case of neuronal 2A cells, it does recover. So we tend to select few mutations, which we know that they can affect the liquid-like property to a solid-like, and then they can cause this behavior. Not only that, now same approach, like I said, we took here as well. We wanted to see that what are they doing, those persistent granules? Do they promote cytotoxicity in the cell? Because the simplest thing that we could do. So what we did is that we induced stress granule, we washed off. Then after some point, we added triggers like amyloid beta peptide or some necroptosis or lytic form of cell death triggers. And we see that some of the DDX3X syndrome mutation, they tend to increase the neuronal cytotoxicity. So what we learned so far is that the DDX3X syndrome mutations can favor persistent stress solid-like granules that can promote neuronal cytotoxicity. We wanted to understand how these DDX3X syndrome mutations alter the liquid-like behavior to the solid-like, because that should explain why this one single mutation can completely change the protein behavior, right? So for that, we wanted to took some molecular approach studies where we wanted to purify the proteins. The problem with DDX3X, it has extended flanking regions, which is difficult to purify. That's why we are a bit truncated, but still retain the flanking intrinsically disordered regions. This is the intrinsically disordered region prediction. And we see that all the proteins along with the wild type we are purified. And then we did a basic biophysical characterization. We see that two of the mutations, which are C-terminal, called 559 and 556, they tend to show altered protein folding than the wild type and the other mutations. And not only that, this particular C-terminal mutation got 556. So the way we purify these proteins these are tagged to the MBP. The moment you cleave MBP, then you can get a purified protein of only that one. You can see that when we performed an assay called MBP cleavage assay using TEV protease, we see that most of the mutations, and including wild type, they tend to stay insoluble. But this 556 protein, it goes more into an insoluble fraction, which is a kind of indication for us that there is the folding defects in this particular mutation. Not only that, we performed thioflavin T assay, which is tend to capture if the protein is undergoing conformational change that is not native-like. You can clearly see that even at 0 time point, 556 tends to show high THT binding than the wild type. Okay. This kind of impression that some of these mutations work can impact the protein folding or stability level. But we wanted to perform a classical condensate assay on a chip or on a slide where we take labeled protein with the RNA and try to see that the kinetics of their condensate formation. You can see that this is a wild type protein without RNA and this with RNA. You can see that their condensate behavior goes up as the time goes up from RNA. You can see that. But when you look at the mutations, particularly the N-terminal mutation, each one has a different behavior, to be frank, because for us, we surprised that all of them have a solid-like condensate formation, but at molecular level, each one has a different behavior. For example, if you take this particular mutation called 182, it is showing enhanced function of wild-type function. You can see that the protein without even RNA can start aggregating. With RNA, the kinetics goes up. But if you look at these mutations, they tend to aggregate even without RNA. It is something which is interesting or also puzzling for us. But the C-terminal mutations are a bit different from the N-terminal mutations. They tend to appear like what I've seen in the biophysical characterization. They tend to disrupt the DDX3X function. That's why the phase separation behavior is way slower than the regular protein. Now, we wanted to understand that now we know that in cellular condition, we have seen that all these 5 mutations, they tend to favor the solid-like granule formation. But when you look at the molecular level, their condensate behavior has been changed. Okay? So we wanted to see that what if it is something associated to its ATPase function, because DDX3X is it's a dead box protein which has a helicase-like domain. Helicase domain, which is having a Recke-like domains. It tends to use ATP and it has ATPase activity because it binds to the RNA. The ATPase activity is required for the unwinding and then separation of protein from the RNA. Otherwise, they would not do that. So we wanted to see that what if the ATPase function can explain their differences in the phase behavior. And then that's what we did. You can see that the black curve is the wild type. To be frank, none of the mutations, what we have tested here, they lost ATPase activity. In fact, some of the mutations have increased the ATPase activity, but they're not correlating with the phase behavior. What does it mean? So if DDX3X retain the ATPase activity, that actually required for the liquid-like behavior of the protein so that it can assemble and disassemble. But the moment its ATPase activity is compromised, they behave more like a rigid conformation because there's DDX3X RNA complex that they don't dissolve much. So we see that the mutations that we have tested, their ATP activity is not altered or they have increased more, and they're not correlating with their phase behavior that we are seeing. Okay? And that's where we wanted to make a point here is that these DDX3X syndrome mutations can activate solid-like granule formation, but albeit because of each mutation have a different effect on the protein function. Okay? For example, here I've indicated here, one of the mutation can alter protein stability and folding, and some of them can enhance RNA-mediated DDX3 condensate formation, or they can promote DDX3X aggregation or homotopic interaction. Okay? We wanted to understand a bit more of it, why each mutation have a differential effect on the protein function, so that they can eventually show same solid-like granule formation. Okay? So for that, we wanted to do certain simulations, real-time simulations, with DDX3X with RNA and see that how their functions are changing. So starting the DDX3X structure from this PDB file, we wanted to start with this, but the problem is they didn't have a complete structure. There are certain coordinates missing. So we used typical modeler, then homology modeling based method to fill up these gaps by filling with the loops. We could not use the alpha fold structure because this is how the alpha fold structure looks like, where all the N-terminals, C-terminals are completely unfolded. And for me as a biophysicist, I don't feel like using this structure because it doesn't give you something meaningful results. And that's why we ended up using this one. Then using this structure, we perform simulation using GROMACS, typically all atom simulation. Okay? It's a large data. Maybe I'll not bore you with a lot of data, but I'll try to show that. In this case, one thing we observed is that if you take DDX3X protein without RNA and start simulating for some time, you can see that the starting structure is this one and the green structure is after simulation. You can see that one of the domain tend to come little downward. That means when there is no RNA, it tend to acquire a conformation which is a bit of a closed conformation, which is a typically expected behavior of the dead protein. So that's what you see here. If you see radius of gyration wild type, it suddenly shifts to one point, like this conformation, and tend to stay there for some time. But the mutations were not like that, especially if you look at these mutations of 182V, 556, and these things. The green one is the wild type. The mutations, they tend to not close completely. That means their conformation is altered because of these mutations in such a way that they don't behave like wild type, and they tend to have a semi-open conformation. Okay? What does it mean? We don't know, but that's what we observed for now. Okay? And now we did same simulation now in the presence of RNA. Here I'm trying to indicate the residues which are required for DDX3X binding to the RNA. That's what we have seen. But one very peculiar feature what we have done, that this is about 500 nanosecond simulation. We even did for 1 microsecond, but it's similarly same thing. You can see that if you see RMSDs of mutations, which are in orange color, they tend to not change their structures at all. That means the mutants, some of these 3 mutations, when the RNA-bound structures are very rigid, unlike the wild-type structure that try to fluctuate or the conformation has been changing. So it's a kind of indicating that their RNA-bound structures are so rigid that might be favoring a solid-like conformation. Okay? Not only that, we looked at all the contact sites and how they're changing along with the simulation. These are the mutants, and this is the wild type, and these are the residues that I have indicated here. The bubble color and the size indicate the occupancy of that particular residue contact sites. You can see that most of these mutations retain more contacts than wild type, except this one, which is going along with that. So let's come back to the same thing. These 5 mutations that we tested, they have completely different molecular properties at each position that they are there, because some of them they try to increase or they make the rigidity firm with when DDX3X RNA complexes. Some of them restricted conformation of DDX3 but not the DDX3X RNA, and some of them are actually affecting the stability of the protein. Okay? Now, considering this concept of protein stability and as well as folding defects, and they tend to more correlate with the protein aggregations that you have seen in neurological condition, which is more like beta amyloid. So we had a crazy idea because it's been known that inflammasomes can cross-seed beta amyloid. We wanted to see that what if DDX3X condensates can cross-seed beta amyloid. We did try. I would not say that we tried in big scale, but we did try some assays where we took DDX3X. First, we let DDX3X wild type or mutants proteins incubate for some time so that they can form aggregation in solution. But take them and sonicate them so that they'll become small seeds. Now take them and incubate them with the beta amyloid peptide. Okay? Then look for how much is the beta amyloid aggregate is happening by using THT, because it binds the beta amyloid aggregates, and you can use the absorption-based stay for that. Okay? You can look at here, these are the curves that we have done when we incubated DDX3X wild type and mutants with the amyloid beta. As a control, we have used amyloid beta cross-seed itself as a starting point. You can see that the TF is the time that is required for cross-seeding beta amyloid peptide into an aggregate. You can see that the beta amyloid aggregate itself cross-seeds, and the time it's required is way lesser than the wild type. But you can see that the 2 mutations, L556 and 559, they tend to show similar like beta amyloid aggregate in promoting beta amyloid aggregation. And we have done some controls also where we perform same assays without the beta amyloid and see that it's not because of the DDX3X aggregation itself. It's typically a beta amyloid. Now, we have seen that some of these mutations can aggregate, especially C-terminal mutations, beta amyloid. Can we say that these mutations, when they promote solid-like granule formations in a cell, because of this beta amyloid-like behavior or the beta sheet-like behavior or unfolding-like behavior, right? So we wanted to recapitulate that in the cellular level. We don't have a proper data. It's just one experimental data that we have, where what we did is that we again did same experiment like what I have shown before. We activate stress granule, wash off, and whatever the persistent granules are not, now you stain them with THT, which is kind of staining to the beta amyloid-like conformation. And this is a score where you see that DDX3 granules, as expected compared to the wild type, these mutations show persistent granules than the wild type. And especially the C-terminal mutation, they tend to show more THT binding, which is kind of an indication that whatever we were looking at the molecular level can be captured here, saying that the principles of folding defects can confer the solid-like conformation by promoting the beta sheet-like conformation in them, and then they can act like a cross-shading points for aggregating more neurological things. That's what we wanted to indicate right now. But to summarize all my data, we identified or we selected few mutations of DDX3X syndrome, which can really impact the protein structure, function, and their RNA binding. And each of them have a differential impact on the mechanism. But all of them, they tend to show solid-like granule formation, and some of them can actually mediate the beta amyloid crossing. Then we try to show them that at the molecular level, they either make the DDX3X RNA complex rigid, or they completely arrest the DDX3 folding itself, and that's how they're causing this solid-like granule formation. So right now, the data we have is, we are here so far, but we want to still understand how DDX3X mediated stress granule formation can cause the neurodegeneration, and with the help of inflammasome. The next step forward for is that we wanted to see that how DDX3X mutations, when they try to induce the stress granule formation and solid-like granule formation, how is it impacting the inflammasome? Or if you take this mutation, are they favoring more of a stress granule formation or inflammasome formation? Because it has a bigger meaning when you look at the brain, because it has it has different cell types like neuronal cells, astrocytes, and microglia. These inflammasome complexes are known to be very well activated in microglial cells, but not in the neuronal cells. And they tend to feed the inflammation in the brain, and they also cause the neuronal degeneration by releasing inflammasome factors. So now we would like to further proceed and see that how these DDX3 variants can affect their functions in the microglial cell level, and start looking at the crosstalk of microglial and neuronal cells. With this, I'll stop here, and thank you. And this is my group, and then we have funding from Indian source, from ICMR, DBT, and the CRB and stuff. And thank you all.
Keshav, you mentioned that some of the mutants have folding defects.
Yeah.
Did you do AlphaFold prediction for those mutants, and what did you see there?
Yeah, we did prediction, and we even ran a simulations on them. We don't see a global change in the structure in the prediction, which is surprising for us, but at the protein level it is. But locally, there are huge differences in the way the surrounding amino acids have been arranged.
Okay, and do the simulated structure and the AlphaFold model differ?
Yeah, the reason we didn't want to depend on AlphaFold is because the moment we put, since these have the intrinsically disordered regions, they are dominating the structure than the actual folders. So we didn't want to depend on that. That's why we took a holistic approach of homology modeling, and then refining the structure with a bit AlphaFold, and then start doing simulations.
Okay.
Yes, it does express good enough in the cells. Like, I didn't show the data, but we have seen that they express well.
No, but they express way too much.
No, we don't say way too much, that without even any trigger, it can aggregate into or stress granules. We don't see that any kind of aberrant expression that can lead to the stress granule formation without any trigger. Correct. I agree with you, because we also thought same thing, and we scratched our head a lot, me and Shivani a lot. And there are papers which recently came that some of these DDX3X cancer mutations, even though there is no change in the ATPase activity, they can alter the phase behavior. If you ask me why, we don't know, but perhaps these are a bit far from the core helicase domains. They may not much affect the ATPase function, but it can be affecting other functions.
With the phase separation.
Yeah,
Interaction with other molecules.
exactly. Interaction with the other molecules and the IDR-mediated homotopic interaction and with the RNA. That's where we are thinking. Yeah.
In your LLPS assay, there were 1 or 2 mutants that did not show phase separation upon RNA addition.
Yes.
So were those mutants by microscopy showed phase or more puncta?
Yes, all the mutations that we tested for in vitro phase separation, all of them formed solid-like condensate formation. And let me clarify which I didn't put the data. We did that only to a certain window of 30 minutes. But if I leave it for 2 hours, even on the slide, and then all of them aggregate. But their kinetics are way different. Yes. Correct. That's where we are coming to the point. One is, I don't want to, I agree with you that it's better always correlate the in vitro data with cellular data, that many other RNA binding proteins also comes into picture. But what we want to claim here is that even though they don't directly enhance the DDX3X function in RNA binding, they're actually diminishing, but their folding is changed in such a way that they have got their own propensity to aggregate like beta amyloid. So that can now completely change their liquid-like property in solid-like. They can still form the granules, and that can be pathogenic or aberrant. No. Close to the RNA binding domain, but it's not.
So under no stress, this is a cytoplasmic protein, right?
Yes, and there are reports which know that it can settle between nucleus and cytosol.
So, I mean, have you tried to see what these mutations do to the function of this protein under no stress? What is this protein required for in terms of RNA metabolism in the cytoplasm?
Exactly. That's the reason why we are doing, it's known, it's well-known role for translation initiation too. So we are doing a polysome from profiling and collecting RNA and see that are these mutations. We are doing that experiment. Overall, what we have seen is that when we looked at the polysome profiling, but we haven't checked at the RNA level each other, we didn't see a great difference, but we need to validate by looking at the genes, and we are doing that.
Hi, Keshav. So I have a question about like DDX3X. Is it a general RNA helicase, or is it specifically involved in translating some special mRNAs?
Ideally, it can, in principle, based on the studies, you put any complex RNA structure, it tend to unwind. But in cellular condition, yes, it's not like it's required for all the RNA translation, but specific RNAs, it's been well documented.
So this is about the dynamics of the mutants. So
Yeah.
do you think that they form faster, and do they need G3BP for their assembly?
Yes, in cellular conditions, yes, they definitely need G3BP1. But in vitro, we can see that independent of G3BP1 also, they can, especially the internal mutation, they can promote their aggregation much faster. And which is known actually because of these intrinsically disordered regions. DDX3 is known to phase separate itself without anything. But in cellular condition, we still think that G3BP1 has a role in.
Yeah, Keshav, wonderful talk. I'm thinking out loud here. So when you see these clinical mutations, have you come across some cases where you have more than one mutation in the same patient? Because like your N term and the C term have opposing impacts. Let's say you have those two mutations coexist. What kind of?
Yeah, we actually looked at, I think so far we haven't come up with a patient which has 2 mutations in one go. No. Not yet.
Since you mentioned this RNA binding activity, was it done in the presence of ATP, your simulations?
No, this is not done with the simulations with ATP. That, yeah, we are considering it, but whatever I have shown was not done with it.
And the binding assay?
The reason we didn't try ATP here this time is that because we didn't see much of a difference with the ATPase activity and all, but I agree with you.
So if there are no further questions, let us thank Keshav.
Thank you.